So today we're going to talk about MHC and MHC restriction. Specifically, we're going to spend a little bit of time talking about MHC genetics, and then we are going to spend some time talking about the structure of the MHC molecule. I will point out that MHC genetics is not always people's favorite thing, and then I once upon a time had a little incident as a grad student drawing MHC, so whenever I talk about MHC structure, I am always afraid that I am going to make another highly inappropriate drawing, as I did then. So if you'll notice, there's one drawing that one thing I'm just not going to draw. I'm just not, because I'm just not. So anyway, remember that we talked about the T cell receptor and the fact that T cells see antigen in the form of a peptide from a microbe that is being presented on an MHC molecule. And so for our T cell to do recognition and make a response, we need to have both the correct MHC and the correct peptide. If the peptide doesn't match, we have no recognition. If the MHC doesn't match, we have no recognition. I told you a little bit last time about the MHC genetics, at least in terms of humans, where I told you about the fact that the MHC genes are on the short arm of chromosome 6 in human. This basically takes up the entire short arm of chromosome 6. And in human, this is known as the HLA complex. So in the HLA complex, were really just random antigens that were on leukocytes that people were having fun, like, making groups of. And so they were called the human leukocyte antigens. We now know that if we look at the HLA, or human leukocyte antigen locus, we can see it's about 4,000 kb on human chromosome 6, and it has 3 different regions. The class I region, the class II region, and the class III region. So HLA is specifically a term that means human MHC. So if I were to ask you something about HLA blah, blah, blah kind, one of the things I would expect is that when you saw HLA, you knew we were in humans, as opposed to when I talk about the mouse thing, that you know we're in mice. So HLA means human. So in the HLA locus, like I said, we have a class I, a class II, and a class III region. The class I region contains MHC class I molecules, which are shown here in red, and they are known as HLA-A, HLA-B, and HLA-C, as well as a few other MHC class I related proteins. They're the ones that are not in red. You don't really need to get super worried about those at this point. In the class II region, we also have 3 MHC class II genes. They're the ones that are listed in blue. And they are HLA, DP, DQ, and DR. You will notice that each of them has 2 parts. The class IIs have 2 parts. The class Is have 1 part. Similarly, the class Is have 1 letter. The class IIs have 2 letters. So those things usually can help you understand what's going on. We also have some MHC related genes in this class II region. And in the class III region, we have some genes of importance in immunology, like all of the genes encoding complement proteins and some cytokines, but they are not specifically MHC genes. Yep?
I was going to ask about the nomenclature of it, but
Okay, so the one thing I do want to mention about the nomenclature, and this is really only kind of one small piece of the nomenclature, is that I've mentioned to you previously that the MHC genes, when we're talking about human, we're specifically talking about the HLA genes. The HLA genes are the most polymorphic genes in the human genome. So if I were to look at all of your genes, you would each have an HLA-A from your mom, and an HLA-A from your dad, an HLA-B from your mom, a B from your dad, C from your mom, C from your dad. And the way we would name those different alleles or those different versions is with a number. So we might call them number A1 or B27 or C15 or something like that. So what that is that means is it's just sort of a list of which version of HLA-A you have of these large number of versions that are available. And as we mentioned last time, this is incredibly diverse. You have very different MHC genes from your mom and your dad, all that good stuff. In particular, if you notice, the most dramatic diversity is seen in the MHC Class I genes, A, B, and C. One of the things that is super obnoxious about MHC is that the original people who studied and understood MHC in humans and the original people who studied and understood MHC in mice did it in totally different ways. As a result, they did some experiments and they came up with some names for what they were looking at. And so in fact, there were two completely different naming systems that were described, which is, as one might imagine, a little bit obnoxious. So like I said, with the human system, it was just sort of this fun antigen on leukocytes, the human leukocyte antigen. In the mouse system, mouse MHC genetics were all done based on skin grafting experiments. And so the experimenters started out with mice of two different strains. One of them you can see is listed here as H2B. Right now we can call that the orange strain. We don't have to quite get into the naming right yet. The other one is listed as the H2K strain. You can call that the yellow one. You can see that this is, because it's an inbred mouse strain, it has both of its chromosomes are the same. They're both orange. Here they're both yellow. And the experimenters could also cross those two mice to make progeny. You can see they have one orange chromosome for each chromosome, one yellow for each chromosome, because they are the progeny of these two inbred strains. Here they are shown as stripy mice. And so what these experimenters were doing was they were doing skin grafts between different mice. And so what they found is that if they took the orange mouse and they tried to take its skin and put it on another orange mouse, it worked. If they did an orange mouse and tried to go to a yellow mouse, it didn't work. And if they went to the progeny, the stripy mouse, it also worked. This one has some orange in it. When they did yellow, yellow could go to orange mice, or sorry, to yellow mice. Yellow could not go to orange mice. Yellow could go to the stripy mice because they have yellow in them. And they further found that if they started with the progeny, neither parent could accept a skin graft because neither parent was stripy. They were one or the other. But you could only see transplant into other progeny. I will first note that this is the outcome that we see in inbred mice. This is not the specific outcome that we see in human transplantation systems because humans are outbred, not inbred. And so you cannot receive any transplant from your parents, as this implies. But here you can see the parent can give to the progeny and to animals that are like itself. the progeny can only give to siblings, not to either parent. Again, things get a little more crazy when we get to an outbred system. But this was how the original experimenters were trying to figure out, they wanted to know just what was this gene that was involved. And so they did all sorts of additional crosses to figure out additional genetic maps. And they were largely caring about whether or not transplants were accepted or rejected, which is officially known as histocompatibility. So that's why it got called the major histocompatibility locus. Because they were also talking about histocompatibility, they ended up calling it the H locus. Somehow it got called the H2 locus. And so just like when you see HLA, you need to have a little voice in your head say, human MHC. When you see H2 in front of something, you need to go directly to mouse MHC. And that's the one that people don't get quite as frequently. So H2 something means we're talking about a mouse MHC, whereas HLA something means we're talking about a human MHC. In the case of mice, this is on chromosome 17. It's about 1,500 kb. You can see that we do have a class I, a class II, and a class III region, although because why not? Class I is broken up, but whatever, not a big deal. We have 3 class I genes in the mouse. They are H2K, H2D, and H2L. And in the mouse are MHC Class II genes are H2IA and H2IE. So again, you can see that we've got 1 letter for class I and 2 letters for class II. So that usually helps in the remembering. Here they're called A and E, but everywhere I ever have seen them written, they're written as IA and IE. So that's sort of straightforward and makes sense. This we can handle. Unfortunately, there's one place where everybody gets super confused when we talk about mouse MHC nomenclature. And it has to do with the numbers I told you about before. So I told you that if I wanted to say, which version of HLA-A do you have, I might like sequence your DNA or whatever, and I might say, you have A12. The way I would like indicate the version is with a number. In mice, we don't indicate the version with a number. We indicate the version in a different way. And it is in a kind of wacky way. In mice, we indicate the version of the molecule with a subscript. And so I'm going to draw this out in a couple different ways, which hopefully will help. Yes, I understand that the way I'm going to draw it is not perfect in agreement with one of the previous slides, but we don't care for right now. So we can imagine first that I have a mouse that is like one of those mice that we use in lab. Officially, those mice that we use in lab were known as C57 black 6 mice. Sometimes just known as black 6 or B6. But that's what we used in the lab. They are completely inbred. So they have all of their chromosomes. They've got 2 copies of the same chromosome. And in that chromosome, they've got a K, they've got a D, they've got an L, they've got an IA, they've got an IE. And it turns out they actually have 2, because they got one from their mom and they got one from their dad. Once upon a time, a long, long time ago, I don't actually know how long ago it was, but whatever, somebody just decided that the kind of K that they have in black 6 mice is called K of B. And so that B subscript is how we indicate which type of the K. Just like we would do A12. Here it's K of B. They also decided that they have D of B, they have L of B, they have IA of B, and they have IE of B in the black 6 mouse. And in fact, since they're inbred, they have the same thing on each. And you can see that here in the C57 black 6, they've got a B for everything. Sometimes we even can use a shorthand for this, because if you look at this, this seems like a lot to write. And so we can use the shorthand for the black 6 mouse and say it is of the H2B haplotype. And so if I say that a mouse is of the H2B haplotype, what that means is that if you drew out its chromosomes, every single MHC Class I and Class II molecule would get a B subscript. And that would be true on both chromosomes. Similarly, there's another type of mice that are very frequently used in research called the Biol mouse. It's a white mouse. So I guess there's my Biol mouse. Somebody once upon a time just decided that its type of all of the Ks, Ds, Ls, IAs, and IEs was D. Why not W? Because that would be too easy. It's just what it is. So this is known as the K of D, the D of D, the L of D, the IA of D, IE of D. I do remember that B is for black, so I can see why you want W. But nope, it's D. And so you can see that, actually, yeah, bulbs are here. They're all D. And so we can even indicate this as these are mice of the H2D haplotype. If I got a mouse and I said, hey, I have a mouse, it is of the H2K haplotype, what would you know about that mouse of the H2K haplotype? Yeah. All of them have the K type. Yeah, so it's K, it's D, it's L, it's IA, it's IE, all are of the K type, and that would be true on both chromosomes. There are situations where you could have like a H2BD. That means it would be the progeny of a B and a D. So you'd have one that's B all the way, one that's D all the way. I'm going to explain, and so that would be, so if it was BD, it would be this. I'm going to explain one more mouse. I'm doing this because sometimes this explanation makes this whole thing click for people. Some people are like, why is she telling us this? This seems super weird. And other people are like, oh, I get it now. So that's why we're doing this. So we can imagine one other mouse here. This mouse would be a mouse that you caught in the New York City subway. We were catching chimps in DSM this morning, now we're catching mice. So we've got a mouse that we caught in the New York City subway. That mouse is not inbred. It has not been sort of carefully bred for lab purposes. And so it does not have the same chromosomes from its parents. It probably doesn't have kind of nice, neat genetics. And so we could go through and we could be like, oh my gosh, this mouse has one version of K that is exactly the same one that came from a black 6 mouse. It has a K of B. It also has a version of K that looks exactly like the one that you find in a Biol mouse, so it also has a K of D. And it might have a D of W, because Mark wanted W, and it might have a D of S, and it might have an L of I, and it might have an L of B, and it might have an IA of K, and it might have an IUD.
What happens when they run out of letters?
It hasn't happened yet.
This was the most polymorphic of genes, right?
Yes, but we haven't mapped every single one in the mouse the way we've mapped every single one in human.
There's like not even 26 of them so far?
I do not know off the top of my head how many mouse MHC types there are. I know that in my life I have used Black 6 and Balb C, and they are B and D. That's what I got for you. But so these subscripts are just telling you of which version of the K or the D or the L that you have, just like the numbers are in the human version. If you look at so I already have a practice problems about MHC. Obviously, you will not be able to answer all of them until we get through the rest of the MHC material, which includes both Friday and Monday. But you can do some of the stuff, and you will see that there are some questions where it's basically asking you questions about MHC molecules in mice or humans. And so you could actually address that information now. So that's kind of the general stuff about genetics. I always feel like that should not be a thing I throw at you right before the exam that's not going on to that exam. Like, it just seems like that's bad timing for that one. So I put that stuff now. But the other big thing that we need to talk a bit about is the function of the MHC protein. So I showed you this very simplified diagram last time where we see that the MHC protein is binding to some peptides that are made in our cell, that are coming from our microbe, and presenting it to the T cell. You had some questions about how that binding works, and we're going to get into some of that detail a little bit more today. One of the big things that you need to remember about the MHC molecule is that the MHC molecule presents linear epitopes to T cells. So these are strings of amino acids that are next to each other in the primary structure of the antigen. So the antigen gets broken down and cut up, and some little string of peptides gets bound to the MHC molecule in its peptide binding cleft and then shown to the T cell. And Friday and Monday are largely going to be focusing on how we break up the antigen and how we get it to the MHC and bind it. So those parts are all going to be Friday, Monday. Here we're thinking more about this sort of structure. Yep?
So in the case of B cells where you have to worry about like the proteins being denatured and the antibody not binding, that wouldn't be the case here?
Exactly. So here, when the antigen is actually being processed to go on to the MHC molecule, it already gets denatured. The cell actually denatures it and cuts it up in the process of putting it on MHC. So yeah, that's one difference that we see. When we look at all of the different types of MHC molecules that we can look at, whether we're looking at K versus D versus L, or K of B versus K of D, or whatever versions we're looking at, the variations that we see really map to that peptide binding groove, allowing for the binding of different peptides. That MHC peptide binding groove will bind to parts of the peptide, while other parts will be available for the T cell receptor to bind. And that T cell receptor will basically be binding to this combined surface of MHC molecule plus peptide together. Last time you guys had questions about parts of the peptide, or how long is the peptide, or things like that. And I was like, not answering any of those. And the reason why I wasn't answering those questions is because the answer actually varies to those specific questions based on whether we're talking about MHC Class I or MHC Class II. And so For the rest of the time today, I have to kind of be zoomed in to either class 1 or class 2 in order to address how this works. There's one other sort of important thing I need to sort of zoom in and tell you. And I was showing these slides to Marina yesterday. I was like, this is what I'm teaching them in immunology yesterday. And when she looked at the next 2 slides, she's like, oh, they're going to freak out about those. And we both laughed. Because you're going to freak out about them, and the things I want you to know about them are really, really simple. And so, yes, you have seen this information in other classes and had to know hard stuff about it. You don't actually have to know all that hard of a stuff about it here. I'm just showing it to you. So don't freak out. So first of all, proteins are made of amino acids. The way that this works is that we have some type of amino acid that has an NH3 on its amino terminus and a C with a double bonded O, and then an O or O minus or OH as the carboxyl terminus. And that's on each a different amino acid. And so when we finally have a protein, our protein always has an N terminus or amino terminus and a carboxy terminus. You can see that the amino terminus has a positive charge, the carboxy terminus has a negative charge. We are linking together many of those different amino acids in order to make a protein. The other thing to notice here is that for each amino acid, we have listed an R group. Those R groups are what make the amino acids different from one another. And these are the R groups. This is the one where Marina said you'd freak out. Here's what I want you to know about the R groups. There are 20 different R groups. They vary based on charge and on shape. When I tried to explain immunology as an undergraduate to my roommate, who was poli-sci? I don't know. She's like a lawyer now. I don't remember what she majored in. Something very not science. I told her that immunology was all about whether or not shapes match. It's actually just like Sesame Street, of these shapes match and these shapes don't. And so what ends up being really important here is that you need to recall that there are different R groups and some idea of the shapes, the shape variation that we see. I will point out that when I am drawing them, I tend to draw either tyrosine or something like lysine, because this one is sort of round and bulky and that one's long and skinny. But that's kind of the difference I'm trying to get at. I'm not trying to like have you know a whole lot of other details. One other thing I just want to point out quickly is you look at this protein, you can see all of the R groups are sort of together off the side. And here we have the amino terminus, some carbons, the next amino terminus, some carbons, on and on and on. And we're going to refer to that as the backbone. We might also refer to that as the main chain when I'm mentioning some things about proteins. So as I said, we need to break this down into class 1 and class 2. Helpfully, class 1 is encoded by the class 1 genes in the class 1 region. And class 2 is encoded by the class 2 genes in the class 2 region. So how we tie this to the genetics is pretty easy. MHC class 1 looks like this. So MHC class 1 includes 2 important proteins. There are really, in total, 3 critical protein chains. One of them is known as the heavy chain. And in the case of MHC class 1, that heavy chain is what is encoded in the MHC region on chromosome 6. So when you saw those genes A, B, and C, those were actually heavy chains. That heavy chain has 3 different domains, or 3 different sort of important regions. So first, we have this one heavy chain that includes an immunoglobulin-like domain and two other domains that are not immunoglobulin-like domains. They all go together. You can see them here as well. We've got our one immunoglobulin domain and our two other domains at the top. We refer to these as the alpha 1, the alpha 2, and the alpha 3 domain. One thing that is really, 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 really important, say if you were going to, I don't know, have to draw this, is that you would want to make sure that alpha 3 has a transmembrane domain. Because in fact, alpha 3 does have a transmembrane domain, allowing this to be an integral membrane protein. The peptide binding cleft is made by this area in between alpha 1 and alpha 2. So this little dash here is the peptide. That's where our peptide binds. And we also have this partner protein that works with MHC Class I. So when we think about MHC Class I variability, this heavy chain, this alpha chain varies a lot. And then this smaller chain, you just have one gene for this. You make a ton of this protein and it pairs with all your other MHC Class I heavy chains. So it doesn't vary. It's completely the same. It's just there to kind of help the structure. And it is known as beta-2M, beta-2 microglobulin. One thing that you should notice about beta-2 microglobulin is that it is its own separate protein with an immunoglobulin domain. It is not covalently attached to the heavy chain. It is also not a transmembrane protein and is not covalently attached to the membrane. So you can see that it is drawn as a separate protein there. In fact, it actually does have an on and off rates from this complex, and there are some fun tricks you can do experimentally because of that. You can see this whole structure here as well. So again, we've got our alpha 1, alpha 2, and alpha 3 with our beta-2 microglobulin. We can see our peptide binding groove in between alpha 1 and alpha 2. You can see it in a ribbon diagram here as well, where you can see that both beta-2 microglobulin and the alpha 2 domain have those nice beta sheets of an Ig domain. And the other two domains kind of have some beta sheets that make almost like a platform, and two alpha helices that make like railings. So basically you have like a platform that holds the peptide and two little railings on the side, so that holds it in between. If we were to now change our view, so right now we're looking at the side of MHC Class I, but if we were to change and now look down at MHC Class I, as if we were the T cell, it would look like this. So what you would see is that the peptide is bound between those two alpha helices in sort of this little region, this peptide binding cleft. One of them is coming from alpha 1, one of them is coming from alpha 2, and then together we have this nice peptide binding region. So you can see, again, that platform with our two alpha helices here. Here you can see the same thing in a space-filling model, where the MHC is in white and the peptide is in red. And this figure shows one of the most important things to understand about the MHC Class I peptide binding cleft. The ends of these two alpha helices are very, very close together. We often refer to MHC Class I as having closed ends. This peptide is really, really constrained in terms of how long it can be. There's no space for like the peptide to like flap out the sides. Like those are super tight together. If you see, the peptide is completely encircled by this. So we have closed ends. The peptide's length is very, very tightly controlled. And so MHC Class I presents peptides that are either 8 amino acids, 9 amino acids, or 10. So 8 to 10. It kind of depends on which one we're talking about. Some will only bind 8, some will bind 9, some will bind 10. But that's it. And they're super tightly controlled in that. So we've got closed ends. only an 8 to 10 amino acid long epitope. Yep.
So you said the white was the MHC.
Yep.
Or the orange was
The peptide. Yeah.
what's
Beta 2M. So that's one really critical part. But there's one other really important part about MHC Class I binding to its epitope peptide. The MHC Class I binding cleft makes interactions with the R groups of the peptides. You can see this here where we've got some dotted lines between R groups on these alpha helices and the R groups of the peptide. You can see another view of this here. Really, your peptide has an N-terminus. That N-terminus is positively charged. And so this whole inside piece of the MHC Class I has a bunch of negative charges to attract that positive charge. This part has a whole bunch of positive charges to attract the negative of the carboxyl terminus. And what you can see is that there are these pockets that will interact with specific R groups of specific proteins. So you might have one that's big and bulky that fits that tyrosine that I showed you. You might have one that's long and skinny that fits the lysine that I showed you. And so they will be differently shaped pockets to accommodate different amino acids. And so what that means is that if you take different MHC molecules, here we call them variant 1 and variant 2. That might be like A2 versus A4, or K of B versus K of D. And you actually pull the peptides out chemically, out of those MHCs, and you sequence them. You might find something that looks like this. So what you can see is that on the top, we've got 4 peptides that got pulled out of the first MHC Class I type. And you can see that at position 2, they all have glycine. At position 3, they all have proline. Whereas like at positions 4 and positions 6 and 7, they got all sorts of stuff. You can see at 9, they have leucine or isoleucine. Those residues where they're all the same are likely the residues that are binding to the MHC Class I pocket. So the MHC Class I variant 1 can bind to any peptide as long as it has G at position 2, P at position 3, or I or L at position 9. Those are referred to as the anchor residues for this MHC Class I molecule. And so when we think about MHC Class I, we see specific anchor residues. When we look at variant 2, you can see it also has an anchor residue at position 2, but in this case, it's a tyrosine at position 2 instead of a glycine at position 2. We also have one at position 9. They could be at any position. 2 and 9 are kind of frequent, but they could be anywhere in that peptide. And so that's how we get the anchor residues. You can also see this in one more way here. So again, you can see this HLA A2 likes to bind to peptides that either have L or M at position 2, V at position 6, V or L at position 9. And so there's one example of something it could bind, but it could bind tons of stuff. B27 likes R at position 2, R or K at position 9. Here's one example of what it binds, but it could bind lots of stuff. Similarly, here's a whole bunch of different peptides all bound by K of B, or a whole bunch all bound by K of D. And you can see that all of them have some residues that are similar to one another. Those would be the anchor residues for those particular MHC types. Yeah, Mark?
So, do different MHC variants have different kinds of binding affinities because of the fact that there's more residues?
So it's not about, so the anchor residues are actually these parts here. And they are changing which peptides will be accepted by that MHC molecule. So as you will see on Friday, this MHC molecule is going to be surrounded by tons of peptides. And the difference for the different MHC types is which peptides they will be able to fit in their peptide binding cleft. There are ways that one can think about T-cell affinity as well, but a lot of that T-cell affinity is based on the other residues here, and so it's not completely an MHC-driven process. So those are sort of the key things about the peptides that are bound by MHC Class I. One other thing I should say about that is that MHC Class I picks up its peptides in the cytoplasm. So peptides that bind to the MHC Class I molecule are peptides that were originally in the cytoplasm. Most commonly, those pathogen proteins are in the cytoplasm because they were made by some virus infecting the cell and being in the cytoplasm. Although any cytoplasmic pathogen protein can be there, usually when we talk virus, we think Class I. Usually there's a link there. If I give you a question on an exam and I didn't tell you if there's Class I or Class II, but I told you it was a virus, if it's a virus, assume Class I. Like they're very closely linked. So one other slightly tricky piece of news is that if you actually look at the T-cell receptor and its affinity for MHC plus peptide, its ligand, the T-cell receptor has terrible affinity. Its binding to MHC plus peptide is pretty weak, much weaker than many other types of biological binding. And so in order for a T-cell to make a proper response, it turns out you need more proteins than just TCR and MHC, because it's not strong enough to hold the two cells together. You need some additional partners. In the case of MHC Class I, the partner is called CD8. And so we have this protein, CD8. It binds to the alpha-3 domain of MHC Class I, which doesn't vary between Class Is, so it can bind to every Class I. While the T-cell receptor is binding to MHC plus peptide, and this stabilizes the interaction so that we can actually get a long enough signal for things to happen. And so MHC Class I always interacts with the T-cell receptor plus CD8. Another way we can phrase that, oh yeah, go for it, Lexi.
I was going to ask,
Yeah.
why is that evolutionarily advantageous?
This means that MHC plus peptide is always going to turn on CD8 T cells instead of CD4 T cells. CD8 T cells are really good at killing things. They're the killer T cell. When they recognize some type of cell, they go ahead and kill it. Well, most of the time, this cell has antigen in its cytoplasm from a virus. And there's nothing you can do to a virus-infected cell except kill it. And so this works out really nicely that we have something from a pathogen in the cytoplasm, probably viral. We know there's something bad in the cytoplasm of this cell, and so we turn on the kind of T cell that will kill the cell. And so this match between CD8 and MHC Class I sort of enforces that function. Does that answer your question? Okay, yeah.
Could it also affect parasitic infections like malaria? The parasite not inside the cell, right?
So Mark wants to know about whether or not MHC Class I affects malaria, parasitic infections.
I mean, not malaria specifically,
Yeah.
because blood cells, just like erythrocytes don't have nuclei, so it
Right.
wouldn't do that in the first place.
So in theory, it could be parasitic infection, but your question brings up one other interesting point, which is which cells of your body have MHC Class I on their surface and are able to present peptides on their surface. So that was a question that you might want to think about. So let's just think for a second. Which cells of your body do you want to have MHC Class I on their surface? So macrophages, what do you say? Why all of them? Because basically any cell can get infected. It turns out that's almost the answer for Class I. Only one kind of cell does not have MHC Class I on its surface, which are red blood cells. Red blood cells don't have a nucleus. So as far as viruses are concerned, they're useless. So we also don't need to have Class I there. But every other type of cell of your body has MHC Class I. So any cell you ever look at has MHC Class I, 3 from mom, 3 from dad, A, B, C from mom, A, B, C from dad, if we're talking about human. Every single cell of the whole body has it. So that's sort of another key piece with MHC Class I. Now we also need to kind of switch gears to think about Class II. Here is an image of MHC Class II. I refuse to draw MHC Class II. MHC Class II involves 2 proteins that are both about the same size. We have an alpha chain and a beta chain. They both have a domain, either the alpha 2 or the beta 2 domain, that is an immunoglobulin domain, as well as more unstructured domains at the top that come together and form the peptide binding groove. You can see that both the alpha chain and the beta chain have transmembrane domains. And so this differs from that structure we saw for Class I. If you look back at the genetics for Class II, you remember there were actually 2 genes for both of them. Those genes encode the alpha chain and the beta chain. And again, we sort of have these nice beta sheets with the 2 alpha helices as sort of cylinders where we're going to enclose our peptide. So big differences here is that we've got, again, we do have 2 proteins, but they are similar in size. The peptide binding groove comes from the 2 of them instead of just being part of the 1 alpha chain. We've got 2 transmembrane domains. One thing that will be really important Monday, that you will care a lot about Monday, some of you who think about biochemical stuff might just think this is interesting today. This complex is incredibly stable. This complex is actually SDS stable. So it is almost impossible to denature Class II plus peptide once it is currently put together. And so that actually has big implications for how it is formed because it is so stable. We can see some big differences in the Class I binding cleft compared to the Class II binding cleft. And so here you can see the Class II binding cleft. And the thing that is important about the Class II binding cleft is that our 2 alpha helices do not come together as tightly at the end. And so we talk about Class II as having a binding cleft with open ends. As a result, Class II is not particularly picky about how big of a peptide can fit in it, because that peptide can flop off the ends. So we often say 10 to 20 amino acids in length. Sometimes it's because like a protease will cleave stuff off if it's flopping too long, but it's really not particularly strict. You can see that peptide binding cleft is coming from the alpha and the beta chain. There are 2 different colors here, and is in the middle. You can also see it's not kind of tightly bound at the ends. In many immunology textbooks, the MHC Class II binding cleft is compared to a hot dog bun. And in fact, in some immunology textbooks, at this point in the textbook, they actually show you a picture of a hot dog. And I don't know why you need a picture of a hot dog, but if you'd like to imagine a picture of a hot dog in a hot dog bun that could potentially go off the sides, you may do so here. So one big difference is how open or closed that binding cleft is, and thus how long of a peptide we can see. The other big difference is that the MHC Class II peptide binding cleft largely makes connections with main chain atoms, those N's and C's that are part of the backbone, not the R group. And so our peptide binding groove is going to make a lot of contacts with those main chain atoms. As a result, what do you think that we can infer about the peptides that are bound by MHC Class II? Yeah, Nick? It's going to bind almost any peptides. Yeah, Mark?
Maybe like something with a large R chain or like proline, for example, might be a bit more difficult to bind to.
Maybe, but in general, we think about them binding just about anything. And so you don't really see anchor residues when you look at the peptides that bind to Class II in the way that you see obvious anchor residues for Class I. And that makes sense because we're seeing binding to main chain atoms, not to the R groups. Every so often, people try to talk about Class II anchor residues, and it's always super sketchy. So like here you can see that all of the peptides that bind to this Class II either have an L or an F or an I at position 4 or 7 or 5. And then also a K or an R or a V or an N at whatever number. And so you can see that like we really don't talk about anchor residues. We really don't think about anchor residues in the same way for Class II that we do for Class I, all based on that binding. One thing that I forgot to tell you, this is actually there is actually a paper that first did this, where somebody first eluted peptides off of MHC and sequenced them. The two main authors on that were undergraduates when they did this. This was their undergraduate project. So wanted to make sure I called that out. The other place where things are a little bit different in Class II is in terms of where Class II gets its peptides from. MHC Class II gets peptides from the endocytic pathway. As we will see more on Friday, we end up having to think a bit about cell biology when we start thinking about MHC Class I. And the cell can be broadly broken down into the cytoplasm and stuff that's across a membrane from the cytoplasm. So there's the cytoplasm, and then there's the extracellular area, and that's across a membrane. But there's also the cytoplasm and vesicles, which are across the membrane from a cytoplasm. Those vesicles are often made by phagocytosis. And so you can see that the stuff that was outside of the cell and the stuff that's in the vesicles are actually connected to one another. They're both across a membrane from the cytoplasm, and they could both be from outside the cell. We have these vesicles that are doing transport throughout the cell. This involves, the Golgi and the exosomes and the exocytic pathway and the endocytic pathway, all that stuff. I don't remember if Dunaway calls it the endocytic pathway, the secretory pathway, or the biosynthetic secretory pathway. But all of them are the same thing depending on which cell biology book you're reading. And in any case, that is where Class II gets its peptides. So usually, Class II is getting peptides from something that has been brought in by phagocytosis or endocytosis. And the peptides are something that are brought in, that are there. And so we see those peptides in an endocytic that are coming from the endocytic pathway, not from the cytoplasm. MHC Class II, just like MHC Class I, has a weak interaction. And so MHC Class II plus peptide also needs a helper in order to stabilize the interaction. In this case, the MHC Class II molecule is bound by CD4. And so MHC Class II presents to CD4 T cells. We are going to see specific interactions between MHC Class II and CD4 T cells. And so another way of phrasing the exact same thing is that MHC Class II plus peptide is the antigen for CD4 positive T cells. So if an antigen gets onto Class II, then it's going to be seen by a CD4. CD4s are really good at making cytokines for other cells. And so we've got a cell that did phagocytosis, and now we might have a T cell come and give that cell some cytokine to improve its ability to do respiratory burst or improve its ability to kill things that it's phagocytosed. And so again, this function ties in quite nicely. But we can also think a little bit about this process in another way. I told you a minute ago that MHC Class I is on all nucleated cells. So everything but red blood cells, all nucleated cells. We can also think about which cells of your body have MHC Class II on their surface. So which cells of your body would you expect to have Class II on their surface? Yep. Neutrophils? Why would you expect neutrophils? Yeah. Because phagocytosis. But now I want you to remember something I told you about neutrophils. Neutrophils are sort of the bad phagocytic cells. They're kind of the stupid phagocytic cells, right? So macrophages you might expect, because macrophages do phagocytosis. Monocytes. Turns out there are only 4 kinds of cells in your body, actually 3 kinds of cells in your body that know how to do phagocytosis. Neutrophils, macrophages, and dendritic cells. And it turns out there are basically 3 kinds of cells that can do a lot of really good Class II presentation. 2 of them are phagocytes, dendritic cells and macrophages. Sadly, no neutrophils. That's because neutrophils are the bad phagocytes. B cells also can present on MHC Class II. And so this is one of the reasons why this stuff ends up being important in B cell biology, is because what B cells do with Class II and how this relates back to the B cell response becomes key later. So only 3 types of cells have Class II on their surface. They are known as the professional antigen presenting cells, or professional APCs. Usually any cell that's presenting something on MHC is known as an antigen presenting cell. These are the professional antigen presenting cells. There are a few other cell types that can sometimes present on Class II. I care that you know exactly one of them. So a bunch of them are listed here. I only care about one. And the one I care about is thymic epithelial cells. So there are cells in the thymus that will be able to present on MHC Class II. That is going to be incredibly important during T cell development. Otherwise, the only cells that have Class II are dendritic cells, macrophages, and B cells. This is also one of the reasons why I always default to Class I. When I'm like, when you have Class I on this cell, because no matter what cell it is, it's got Class I. If it's Class II, I actually had to think. The last 2 slides here are summary slides comparing. So you can see that we can compare Class I and Class II, both which parts of the proteins make the peptide binding cleft, either alpha-1 and alpha-2 versus alpha-1 and beta-1. Is it closed or open? How long is the peptide? You can see that they give a slight different answer for Class II, because really, Class II, there's so much variation. Whether we have anchor residues, whether the peptide, how the peptide is bound. You can also see a different view where we can compare and contrast the structure, which cells have them, which T cells respond, and where they get the peptides. On Friday, we are going to go through the process of Class I getting its peptides. On Monday, we're going to go through the process for Class II. On Friday, we will have to spend a little time reviewing a teensy bit of cell bio, because it's going to come up in a lot of this. So be aware of that. And yeah, I will see you guys in lab.